and welcome to this new episode of Cytobytes. I am Serena Di Cecilia, and today we will talk about clustering QC. Clustering is the most powerful tool in high-dimensional flow analysis and mass cytometry analysis. But after the algorithm finishes running, we should always ask ourselves, can we actually trust the results? We clean the data, we run UMAP, we run TSNE, and then FlowSOM, and then generated beautiful clusters. Everything looks great. But before we QC our clusters, we should remember why we cluster in the first place. Dimensionality reduction is a map, not a population definition. And so a region on a UMAP or a TSNE may still contain mixed phenotypes. So why QC after clustering? Because beautiful clusters are not always biologically meaningful. One of the common mistakes is to stop after algorithms produce the colorful clusters. But if QC is skipped, clustering can either split one population into multiple ones or eventually merge distinct populations together or create results that are hard to reproduce. So the bottom line is always validate before interpreting. So what does good clustering mean? A good clustering result is not simply a nice visualization, right? We are looking for mainly three things. The first one is stability. So would a similar structure appear if we repeated or slightly adjusted the analysis? The second thing to look for is separation. Are clusters distinct from one another in the multidimensional space? And third is the biological interpretability. Can we actually describe the cluster using marker expression and cytometry knowledge? A strong result is therefore compact, separated, and biologically meaningful. Today, we will use three practical checks, which are the FlowSOM outputs, ClusteRCheck and Taylor index statistics, and some extra tools to evaluate our clustering results. So the first level help that we can get is from the FlowSOM metaclusters heat map. FlowSOM is an algorithm that first organizes cells into nodes and then groups them into metaclusters. At this stage, what we can do is to look for coherent marker profiles, right? So that is a good sign. If a metacluster represents a real cell population, then the expression pattern should be explainable, so it should have specific markers to describe that population. But if multiple metaclusters look even small, nearly identical across all the markers, probably these may indicate some overclustering. And we could think of something like this, like in the second and third row of our heat map right here. So what do we need to do in practice? In practice, we need to flag those metaclusters and compare their marker expression. And eventually, we can consider reducing the number of clusters before moving on. The second output that we can get from FlowSOM is the minimal spanning tree. This tree is helpful because it shows relationships between clusters in the high-dimensional space. Distinct cell populations often appears as neighbors or branches. If different biology is collapsed into a single node, then the solution may be that there is a lack of resolution. Vice versa, there could be some adjacent nodes that have almost identical marker expression, and the data may be over-fragmented, basically. In this example below, when we input the number of clusters into FlowSOM, we first initially inserted an input number of 8. And as you can see, there is this purple cluster, which is the cluster number 7, which seem to have different subpopulations in it. So some clusters looks like they collapsed into a single node. But then when we increase the number of clusters to 15, there is actually a resolution of this unique cluster into multiple clusters. A different view from the actual minimal spanning tree is also available, and this can help even more to see if there are under or over clustering results. Like this one, for example, is the output from our 8 input number of clusters, and you can see that the nodes are not really nicely resolved, but when we increase it to 15, we have a better visualization and a better resolution of the clusters themselves. If you want to know how to define the input number of clusters into FlowSOM, there is a video that Sophie made about that on our Cytobyte channel, so just check it out. Another way to inspect our clustering results, and specifically under clustering, is by using ClusterCheck. ClusterCheck asks whether a cluster behaves like one population or whether it may contain multiple structures or hidden substructures, let's say. It checks basically the modality across parameters, and in simple terms, what it does is basically checking whether a marker distribution looks like one peak or can contain multiple peaks, as you can see in this example here for CD8 and PD1. So if one cluster shows high modality for important markers, probably that cluster needs to be inspected manually, and you can do that by using indeed these histograms. Please keep in mind not to only check for modality on the UMAP or t-SNE view. ClusterCheck is way more informative when you look across antigen expression parameters that define the biology, even simply by using histograms, as you can see here. So what should I do? Well, if you see high modality in one or more markers, inspect the markers histograms and the location on the cluster or UMAP or minimal spanning tree. Then ask whether the pattern corresponds to a clear biological split. If yes, refine the clustering or split the population. If not, keep the cluster but annotate it as heterogeneous and avoid overinterpreting. So basically, deep dive. The other statistic that I want to talk about is the Taylor index. The Taylor index statistics instead shift the question from what is happening inside one cluster to how pairs of clusters relate to each other. The matrix that you see here shows comparisons between clusters. It compares basically the compactness within clusters with separation between clusters in the n-dimensional space. That means that larger values, so lighter colors, point to clusters that are better resolved, while smaller values or darker colors suggest poor separation or high similarity between cluster pairs. So what's the final workflow? What should I do? How should I investigate this post-clustering QC? A simple QC sequence is to first inspect the FlowSOM meta-clusters. If you're not using FlowSOM but you're using Phenograph, just as you know, there is a heat map that comes out of Phenograph as well. Or if you're using XShift, you can use additional tools within your platform. For example, in FlowJo, we have Cluster Explorer. Then you can review the minimum spanning tree neighborhoods, as I showed you before in the different views, and realize if there are multiple substructures within the same cluster. Then you can use ClusterCheck to evaluate underclustering and Taylor index to evaluate overclustering. So to see if multiple populations that are basically belonging to the same biological population are split into two different ways. As a note, ClusterCheck and Taylor index are available in R and also in FlowJo. And then once you've done that, you can go on to the last step, which is the phenotyping. Phenotyping, probably there will be another video about it, but for the moment, you can phenotype your clusters by using all the different commercially available platforms or the tools within the platforms. I can suggest that there is one platform that is called Cluster Explorer in FlowJo that helps phenotyping a lot. So the final decision, once you have done all this check, is usually to merge redundant clusters. So if you see clusters that are similar and they belong to the same biological population, you can merge them. If you see that multiple populations are merged into a cluster, you can still split or refine the number of clusters into your clustering algorithm. And then if you see that you have a specific cluster that does not fall either in the underclustering or overclustering solution, then you can annotate it, but keep the limitation, keep the information. So the takeaway here to keep in mind is that clustering is not the endpoint of high-dimensional cytometry analysis. FlowSOM outputs, phenograph outputs helps us understand the structure. ClusterCheck helps us identify clusters that may contain hidden heterogeneity. The Taylor index helps us to evaluate separation between clusters, and all of this together can help us to make a decision based on marker expression and biological knowledge. Thank you so much for watching, and I'll see you at the next episode of Cytobytes.